So we've used high-resolution CT scanning to look inside the brain cases of a group of dinosaurs, and we focused on 2 specimens from the UK, Baryonyx and Ceratosuchops, and we reconstructed the brain and associated soft tissues in order to figure out how they're interacting with their environment. Well, we found that the overall shape and olfactory and hearing capabilities are very similar to other carnivorous dinosaurs, and this is slightly unusual because spinosaurids are semi-aquatic to a degree, so they're probably hunting fish and other aquatic prey, and we were trying to see whether that unusual ecology was reflected in the morphology and sensory capabilities of their brain and associated tissues. And what we found is actually they're very similar to other large-bodied theropods.
From this work, we have a couple of ideas, hypotheses about what might be going on here. Maybe spinosaurs are pre-adapted with features that they've inherited from their theropod ancestors to be able to take advantage of an aquatic environment and fish from those habitats. Maybe all they needed to do was elongate their jaws and develop teeth rather than the blade-like teeth like T-Rex and allosaurs. Maybe these simple points for catching and grasping fish, and maybe that was enough. Another idea, which might not be entirely separate from this one, is that we've basically managed to reproduce the space, the cavity inside the brain case that the brain was sitting in. And so we haven't managed to highlight specific features of the brain which may have been enlarged or developed to allow animals like spinosaurs to catch fish, and so we're just not seeing the specifics. So the gross shape is no different from other theropods, but the specifics, the smaller parts and structures in the brain which may have changed, we can't see. So we've still got work to do. CT scanning is so useful across so many different fields, but for paleontology in particular, we can now get inside fossils in a way that we never could before, and we can start to reconstruct the soft tissues. We can start to see features that are embedded inside bones that we would never have been able to see without destructively sampling bones before. So we can see all kinds of new and exciting structures, and we can start to really understand fossils in a completely new and novel way.